So now we get to the really mathy part of the morning. I'm joking. I am pretty far from a statistician, pretty far from a mathematician. A little bit about me, um, and then I'll talk about kind of what the overall point is. My graduate work was in microbiology and immunology. I moved into bioinformatics kind of in as a postdoc because some of my graduate work, and I have been self-trained bioinformatics for about 20 years, but really it's because of the great people that I work with, the statisticians, the other bioinformatics people, the software developers who've really, who I consider friends and resources, right? I'm like, I don't know how to do this test. I don't know if the test is appropriate. I go to people like Lisa and Rachel and Kelly and ask those questions. So there may be things that are said in here that make the statisticians gasp. I hope not. I'm not going to show you, I think, any equations. I'm going to talk kind of about what we do with this data, right? So you've taken, you have a study where you've generated 10,000 transcriptomics data sets. And this is common for, especially for bench biologists who are like, we got a grant to do transcriptomics. And now I've done 200 transcriptomics data sets and it's crashing Excel every time. Like, okay, so we've already gotten past that point, right? Excel is probably not the tool that you want to be using to analyze this kind of data. But really, after you get through the very important steps of getting to the point where you have differentially expressed genes, and I should note that I'm talking about transcriptomics today, so I'll be talking about genes, but you might hear me say proteins from time to time. I work a lot with proteomics data. A lot of the stuff that I'm going to be showing here is actually applicable to anything that can be mapped to a gene, gene products. So that would be proteins. We also work with phosphoproteomics. We work with some other kinds of genomics data that can be mapped back to a gene identity. So really the point of the post-differential, or I say post-expression, post-differential expression analysis, you've gotten to the point that Lisa talked about. You've got a very large pile of data, and you're at the point where you're like, okay, so I've got a bunch of differentially expressed genes. I've done all the right things to get to these steps. What does it mean? So in the example that I just mentioned with Excel spreadsheets, I mean, this is totally not a joke in that oftentimes when those are delivered to a bench biologist or someone who's initiating those experiments, they'll just do a sort on differential abundance and they'll go, gene by gene through and say, oh, I know that one. Not sure what that one does. I think this one does something in this pathway. I think this one does something in that pathway. So what I'm going to talk about is a couple of different approaches for kind of organizing that process. How do you take this big pile of differentially expressed genes that you've generated, or multiple piles of differentially expressed genes, and make some kind of biological sense out of it? So oftentimes the experiment is telling you something. You're experimenting to identify the effect of temperature on CO2 production in your favorite bacteria or something like that. And you've got a bunch of different temperatures, and you've cultured this bacteria under different temperatures, and you've gotten differential gene expression from each one of those. And you want to know something about the large scale, like what's going on, what kinds of pathways are active, because you want to know something about the phenotype and the mechanism of what's going on under the hood, right? That's why you're doing these experiments, to kind of get a big picture idea of what's going on. Obviously, it's really important if you're looking at individual genes, you want to know what those are doing too. And that's kind of where we've gotten to in the past talks, right? We've gotten to that point of like, okay, I identified this list of interesting genes because they're all upregulated when there's a high temperature, for example. So really it's, how do I query this big pile of data and figure out how to extract biological meaning mechanisms, and further hypotheses for further investigation. So the structure of my talk, post-differential expression analysis, I would lump visualization into this. Lisa's already covered this kind of with heat maps, PCA, some other kinds of visualizations where you're trying to get like a visual idea of the patterns in the data, especially when you're looking over a large number of different conditions. Again, that example of temperature variation, a heat map like that might show you, here's a block of genes that's all upregulated in low temperature, and here's another block of genes that's all upregulated in high temperature. And you can visually see that there's patterns in your data. I'm not going to talk about that because it's kind of been covered. I'm mainly going to focus on two separate but related approaches. And this isn't exhaustive. There's many different things that people do at this post-differential expression piles of genes. But one you'll see over and over and over again is pathway or functional enrichment. This is statistical enrichment in pathways. And I'll talk in a minute about what we mean by pathways. But the idea there is that if you are looking in your big pile of genes that are differentially expressed, and you can see that there are certain pathways that are overrepresented, then that's a good indication that whatever that pathway is doing, the function is being carried out at that time, right? So transcriptomics is a way to get at those questions. There's a couple of other things you can do with enrichment approaches. I'll kind of talk a little bit about that, and I'll talk about some of the most common ways that you'll see for pathway enrichment. If you've done pathway enrichment before, or if you're really well versed in it, this is all going to be review, and it probably at a pretty high level. But hopefully you'll get some interesting information out of it. If you haven't done pathway enrichment before, and you've read papers in transcriptomics, you've definitely seen pathway enrichment used and fairly useful. Another thing that I'll touch on is it's related, it's network analysis. And this is a way that you can represent the differentially expressed genes, or the genes that you're getting out, as a network or a graph. And there's a couple of different ways to do that. One is by using existing knowledge frameworks, protein-protein interactions, other kinds of associations, and kind of combining that with your differential expression. And the other way, which I'm going to talk a little bit more about, is you can infer associations using statistical patterns, or using statistical associations between the patterns of the genes that you might have. If you're, again, the example where you're looking at a variation of temperature, you can see which genes are co-affected by temperature in the same way. This would be something like what I was explaining for the heat map, but we can get a little bit more detailed with it if we're looking at a network approach. So before I get into the enrichment approaches, what do we mean when we say pathway? Pathway is one of these loaded words, loaded terms that kind of means some different things to different people. So here's some examples of the kinds of things we might mean by pathway. One thing I'm going to preface this with too is that some of the examples that I'm going to talk about draw from kind of human health related, so cancer related studies. And I'll try to kind of delineate where those are applicable and how they might apply to kind of prokaryotic studies, bacteria. So in the upper left here is a pathway. This is a signaling pathway from a cancer study we did about 4 years ago. This is actually an updated version that we put out for a review. And it's basically a signaling pathway is something where you're taking a signal. In this case, it's DNA damage. This could be from inside the cell. Oftentimes it's from outside the cell. Something is signaled on the outside of the cell. It gets translated into a signal by some receptor. And then the signaling pathway is actually a transfer of information from this signal down into an action. In this case, we were postulating that it disrupts cell cycle. But a lot of times it ends in some activation of a transcription factor and upregulation of a bunch of genes that might do something, a stress response, for instance, or some other kind of action that the cell or the organism might want to take. And so signaling pathway is something that you'll often hear, especially in disease, human disease. Pathway is almost synonymous with signaling. On the right-hand side, this is a high-level overview of metabolic pathways. So metabolic pathways are a bit different. They involve proteins, gene products that are generally enzymes. As input, they take some set of small molecules that they then transform to an output, which is another set of small molecules, right? So this could be taking some kind of larger molecule and breaking it down to provide energy, or it could be taking a collection of small molecules and putting them together to build something bigger. And so metabolic pathway is another place where you hear pathway, but they actually have slightly different meanings, right? And then a third place that you might find this is something like, I have an example of photosynthesis from the KEGG database. And this is a series of complexes where as input, you're taking light energy, and as output, you're making chemical. And so this is even another kind of sense, not information, not explicitly small molecules, it's energy, electrons. And pathways are sometimes, you'll see a pathway database that has ribosome as a pathway. Is ribosome a pathway? It's a big complex. It does something. But a lot of times those are kind of used as loose synonyms. And so the last one I wanted to throw in here, because some of the databases that I'll talk about in the next slide have other things, bag of genes that kind of does the same thing. Stress response is a great example. So as stress response pathway, you can imagine that there are stress response pathways that look a lot like this. In fact, there are eukaryotic stress pathways where there's some signal like damage, where it's transmitting information down to some action. Then after this action, there's a bunch of things that happen. All these genes get upregulated based on this input. And those sometimes are treated as separate bag of genes. They're not maybe communicating with each other, but they're all kind of pointed in the same direction functional. And so it's just something good to remember, is that when you're looking at the output of pathway enrichment, those things can actually mean different things depending on what kind of pathway database and what kind of pathways are being enriched. And I'll try to give you a couple of examples as we go. So I just wanted to put this here as a reference of some kinds of pathway databases that you've probably seen, or you've seen at least one of these, you're aware of. And I have a couple of columns here. One is whether or not this is very applicable to prokaryotes. Most of these, maybe not all of them, most of them do have prokaryotic annotations included in them, prokaryotic pathways included in them, but many of them are not centered on prokaryotes. David is a good example. It's from the NCI, the National Cancer Institute, so it's barely focused on human. And then the other one I have here is something I'm calling canonical, which just means, does this database represent things that are kind of distinct units where most of the functionality is incorporated in one thing, and it's not spread, like you don't have multiple things that are all talking about the same function, for instance. Gene ontology, and I won't talk in depth about all of these. I'm just going to go over a couple of them. Gene ontology is something you might have heard. It's an ontology, which means it's like formalized way of capturing functional labels for things, and it kind of originated from the eukaryotic side, but it's been translated, or other versions of it have been done for bacterial systems. And it's basically a hierarchy where the different pathways or the different groups are at different levels. So you can have things as broad as enzyme, right? Is a protein, what's a protein's function? It's an enzyme. Somewhat useful, probably not useful if you're trying to interpret your big pile of differentially expressed genes, right? And then below that, you have multiple different kinds of enzymes. So you have metabolic enzyme, you might have kinase, you might have, something else. And then below those, you have subdivisions. The reason I'm telling you this is because if you are getting output functional enrichment from gene ontology, you'll oftentimes get all of these things, right? You'll get enzyme being upregulated, you'll get metabolic enzyme being upregulated, you'll get serine threonine transferase, whatever, being upregulated. And it can be difficult to kind of judge what level that you should be looking at in terms of interpretation. Other databases, KEGG, which is the Kyoto Encyclopedia of Genes and Genomes, is fairly comprehensive, so that's nice. They've taken care to do a lot of these different organisms and annotate them. And it has more kind of canonical. You'll look at them and you're like, oh, it tells you it's a signaling pathway, and that signaling pathway, their genes pretty much appear there, and they might be shared with a couple of other pathways, but you're not getting a lot of kind of multiple overlapping things, and easier to interpret. The other one that is kind of primarily originates from prokaryotes is called COG, which means clusters of orthologous genes, where they basically, orthology is just based on the sequence similarity between two genes. So if you have a gene in this organism, this bacteria, and a gene in this bacteria, and they're very, very similar in terms of their sequence, then you can group them together, and then functional labels can be applied. This is some kind of kinase, for instance. That originated in prokaryotes. It has been extended to you. Some of these other, many of these other databases have like very similar kinds. Like you'll see that Reactome has a lot of the same kind of metabolic pathways as KEGG. They have other things. So in terms of which one to use, it's really a matter of kind of which one seems to be most tailored to what you're looking at. And if you're looking at things like metagenomics, that can be difficult, right? So you're looking at a lot of genes that really, or a lot of organisms that you really haven't, there hasn't been laboratory studies done on them. There hasn't been big annotation efforts. So a lot of times what you're looking at with these are computationally transferred annotations, which are reasonable, right? They're based on orthology and things like that. But you're not going to have a specialized database for your weird bug that you just isolated from. So the purpose, or the way that that pathway enrichment is done, this is very similar to kind of the stuff that Lisa was talking about with differential expression. You're basically looking at differential expression of a set of genes rather than individual genes. And I use this this analogy of surprise, right? How surprised am I that I see my pathway in my set of differentially expressed genes? The set of differentially expressed genes is this big pile of rocks here, right? Got a bunch of black rocks, got a bunch of red rocks. How surprised am I that there's this number of red rocks in this pile? This is your differentially expressed. How would you answer that? If you just had to answer it right now, you don't know, right? You have no idea because you don't know how many red rocks there are in the whole group. So that's an important thing. Your background is important. And basically the tests that I'll talk about, the kind of approaches, are based on this idea, which is like, how surprised am I to see these red rocks representing the pathway that I'm focused on right now are distributed in my pile of interest. So if you look in the back of this crudely drawn dump truck, who is the artist here? What I'd like to know. You see, in one case, you're not surprised much at all. You see about the same distribution. You're like, okay, the dump truck just dumped those differentially expressed genes off whatever pathway of interest or pathway that I'm looking at right now. It's not any different. You're not surprised. In this case, where most of the rocks in the back are black, you'd be pretty surprised, right? You'd be pretty surprised to see that if a dump truck just randomly dumped stuff off, that you'd be seeing that. So the surprise is often expressed in a p-value. So there's a couple of different methods that I'll kind of briefly go over. You can look further into these. And these represent kind of, I would say, classes of methods rather than individual methods, although I will mention an individual. So that method that I just talked about, where you're basically just counting the number of rocks, that's something like the hypergeometric test uses a Fisher's exact test, or sometimes there's other names that are given to it, but it's basically looking at set overlap. All you've done is you've taken your differentially expressed genes, you compare them with your non-differentially expressed genes, and then you say, for my pathway that I happen to be looking at right now, what's the distribution there? And you can do that, and you can calculate a p-value to say, wow, this is super surprising that we see 15 members of this pathway in my differentially expressed pile. And then the other metric that you'll get out of that sometimes, and it depends on the method, is something like an overrepresentation metric. And that's just like, how far away from the mean are we? Like if we were just looking at randomly pulling out rocks, how many times would we pull out? If you have 15, maybe randomly you would only expect to see 5, and so you see an enrichment of 3. So that's kind of just a general magnitude of difference. So you get those two values, p-values and magnitude of difference. So the other method is something that has various names. It's been implemented in a number of different approaches. Probably the best known is gene set enrichment analysis, and this is from the Broad Institute. And this is where you take something like the differential abundance for your sample and you rank. You don't make a call about whether or not something is differentially expressed or not. You don't threshold. You just take all of those differential expression abundance values and you rank your genes from most to least. And then you basically look to see, am I surprised for my pathway of interest at seeing the clustering of is my pathway of interest randomly distributed in this list, or does it seem to be non-randomly distributed? So the idea is very similar to the simpler kind of set enrichment here, but in this case, you're kind of using more of the data. You will get a p-value out of the other side, and you also get something, GSEA calls it an enrichment score. It's or a normalized enrichment score, so you'll see that as well. And that basically gives you an indication of how far from middle of the list are you, basically. Are you way on this side? Are you way on that side? And it kind of correlates with the p. So I meant to say this at the start of this slide, but the way that these approaches work basically is you say you have a differentially expressed set of genes and you want to use one of these. You input your differentially expressed set of genes. You, depending on the tool, either also input your background set. What was this drawn from? Or you select a background set. David, for instance, has background set select. And so generally your background set with transcriptomics is everything that you measure, right? So all of the genes that you had a measurement for is your background, and the differentially expressed is your set. And then what the algorithm does is it basically says, now give me a database of pathways. And this is something like KEGG, right? And it steps through every single pathway and it does these tests. And then you'll get metrics out, you'll get p-value and some kind of fold enrichment for every single pathway. Now, if you've been listening closely, you know that what you also need to do at that point, after you've done these 2,000 tests on your same data set, is that you should, and most of these methods do, implement some kind of multiple hypothesis, which is basically to correct for the number of times that you've tested your list, right? And so that can adjust those values down. Right, so that's all I wanted to say on that. I have a list of some of the tools in a couple of slides. So how do you interpret these enrichment results? And I think I've kind of given you some idea of this. And different tools will kind of have different filters and different ways that they represent things. These are just things to be aware of. One of them is to look at how many genes are represented. If you get a pathway that has a really great p-value and you kind of take it as is, what I've found is that sometimes that really great p-value reflects 4 genes in your differential expression. You're like, is that really what I'm interested in looking at? The pathway has 20 genes. I only see 4 of them in here. For whatever reason, it gives me a very good p-value, but maybe that's something that I want to take, I want to take a little bit closer look at. So it's always a good idea to take a look at the number of genes and the kinds of genes that are being represented in those pathways when you get the image. Another is whether or not for your experimental setup and your questions, do you want to consider negative enrichment? So in this case, this would be, I'm very surprised to get this result. I don't see any members of my pathway and I would expect to see 5 members, right? So that's interesting, but is it really that actionable? You're looking at differential expressed tile and you say, here's all the pathways that are underrepresented. There's some utility in that, but generally I find that most questions you're asking not as interesting as the things that are positively enriched. Consider the p-value, how surprised, and the magnitude of normalized enrichment scores, or some kind of fold enrichment. I think I emphasized this before, know the kind of pathway database that you're using. Most of the tools that I'll talk about either have their own pathway databases that they constrain you to use, at least in the web and the web versions of them, or they allow you to select from pathways. And mind the background. Again, with transcriptomics, your background is generally everything that you've observed. With things like proteomics or other kinds of comparisons that you might make, the background gets to be pretty important and it can actually be a thorny thing. So sometimes just the choice of a background can make certain pathways seem really enriched when that's something just to be cautious about. This is an example, again, from proteomics and phosphoproteomics, very similar. This is a very standard way that you'll see things represented, maybe not with the bar graph, but representing the p-value as a negative log p-value, negative log 10 p-value. So essentially that means the smaller the p-value, the larger the bar, the more surprised you should be. So in this case, these pathways on the left are all very upregulated or overrepresented in our data set, and then they kind of tail off. So some of the questions, and I think we've gone over this already, but some of the questions you might be asking are the same kinds of things you might be asking with differential expression. But now where you're asking it at the pathway level, you're a step closer to kind of that phenotypic output that might be more biologically actionable. What are the pathways active in condition A versus some control, or in condition A versus condition B? What are the pathways active in individual samples? So that example that I gave where you have a bunch of bacterial cultures and you're varying the temperature, maybe you want to know for really high temperature, what are there individual pathways that are more active? This is important when you have, I would say, more individual kinds of samples. So if you're looking at patient samples, for instance, you might be very interested if a cancer patient has a super high activation of a particular pathway, even if the group that you put them in doesn't have that, right? So you might want to look at it, individual samples. What are the pathways active in a specific subset of genes? And so that heat map example that Lisa was showing before, where you get clusters of genes and you can see the overall pattern, but you might not know what they mean because you don't know what all of the labels are for all of the different genes you see. Pathway enrichment can be one way to do that, right? You get a bunch of genes and they're all activated in the high temperature treatment. And then you can do pathway enrichment on those genes, get an idea of what that cluster means. So that's something that's commonly done. With a heat map, you'll see functional pathway labels next to that heat map to say, this block of genes has stress response, it has heat response pathways, it has these other things that might be more exciting and more interesting. And the other thing is, what pathways, and this is something you well, what pathways correlate over a range of samples with a measured variable. This might be if you're judging carbon dioxide production based on temperature, you have carbon dioxide measurements for all these different samples. What are the pathways that are most correlating with carbon dioxide production? And hopefully what you find is that you find some things that you know about, like carbon dioxide production pathways are correlating with carbon dioxide production. And then you also may find some interesting things that you didn't know about before. So I've mentioned a few of these, and these are some resources you can take a look at in this. So David is again from the NCI. It is fairly commonly used for kind of human health related things. GSEA, the gene set enrichment analysis, is through the Broad. They have a nice interface. I think they have a web interface now, and they also have a Java app you can use. Enricher is one that I've started using more recently. It provides just a really nice, it's a web interface that just provides a really nice set of options for doing kinds of pathway enrichment. You can choose different pathway databases. And then it also provides a whole bunch of different kinds of plots and things that you can use to kind of understand your data. And what I've found is with Enricher specifically is all those different plots, when you start to understand what they mean, it's really useful to go in and say, oh, this, I thought this one thing from just this one plot, but if I look at these other plots, maybe there's some other story that might be more. Web Gestalt is a little bit similar. And again, this is a non-exhaustive list. These are both ones that are very popular, I would say, and ones that I happen to know about and use. And then the last one is just a shameless plug because this is our library that I've built. There are lots and lots of different R libraries that will allow you to do different parts of this. GSEA has an R library that you can do this. So I don't want to say that ours is the only by any means. It is. It does basically fairly simple kinds of comparisons, but it incorporates those different kinds of enrichment that you can do all in one place. And it's also targeted at multi-omic data integration. So the multi-omic kinds of talks that you hear tomorrow, this is a tool that where we're specifically trying to design it to be better able to incorporate multi. So I want to spend probably not too much time on network analysis. Network analysis, again, I mentioned there's kind of two different flavors that you'll see commonly. One more common is using existing knowledge frameworks to understand your data better. And this would be things like protein-protein interaction. I don't know if you've all seen those hairball protein-protein interaction networks where everything's going in all different directions. So you can kind of argue, is that actually very useful? From a visualization standpoint, if I plot my differentially expressed genes on top of this hairball, is it going to tell me anything? Maybe not. But there are other knowledge frameworks that are actually pretty useful, and that can be to identify things like, okay, we've got the set of the set of differentially expressed genes. We put this into a network where all of these different relationships, and you can start to see things like, oh, now we've got a set of differentially expressed genes that are all linked to this transcription factor. Well, that might mean that they're all regulated by the transcription factor. So it provides hypotheses to go forward. And I'll give you some examples of tools you can use to do this with your data after this. The other way is probably less well known, but I have experience in using it, and it's something called network inference. And this is basically where you're taking those patterns of gene expression in your data over multiple different observations, and you're using those patterns to connect individual gene relationships, right? Those relationships are inferred, so they don't necessarily mean anything physical. They don't mean like that things are physically interacting. They don't mean that they're co-regulated, but they can be useful, and I'll show you a couple of ways that they can. And one of the ways has to do with topology. So talk about that. So this is an example where we used a network inference. This is a very simple network inference. We basically took, and I'll explain what the experiment is, but the network inference part is basically just correlation. We took Pearson correlation of gene expression over time, and we basically used a threshold, and we said everything that's more correlated than 0.9 will draw an edge between. So these dots are individual genes. The lines between them are correlation. I think it was above 0.9 or maybe even higher. The experiment is a cyanobacteria that has a circadian rhythm. It photosynthesizes during the day, light, and then it does nitrogen fixation at night. Those two processes are basically mutually exclusive in a chemical sense. And so it does this all in the same bacteria. So the same individual does this. And so it has a very tight regulation of its gene expression profile. This is from a study that we did over a 48-hour time course where I think we had 36 different transcriptomic data sets from this cyanobacteria, and it was just a diurnal cycle. So it's basically light dark cycles that it's growing in regular media. And so we were really trying to capture what were the patterns that were happening of gene expression over time in these light dark cycles. Now, the interesting part is this is a great example of where you're using something like network inference. With the modern methods, you might not even call this network inference. To really visualize interesting things that are going on in this cyanobacteria. I'll explain very briefly what this kind of means. So all of the genes that you see on this side basically have peaks during the light cycle. So they are peaking during the light cycle. They're basically on some kind of sinusoidal rhythm, right? By their light and their circadian rhythm. And all of these are peaking in the dark. And when you do pathway enrichment on sections, so basically what we did is we clustered these on the network into sections, and we did pathway enrichment, and not surprisingly, photosynthesis pathways come up here, but we also saw carbon fixation. This is a very high level summary. So we saw a lot of different functions in here that were interesting. And then as you transition to dark, you get a transition to metabolism. And again, that's a very catch-all, right? Like a lot of things going on there. But nitrogenase was the important thing right here. And then you get a transition at the dawn back into this photosynthetic. And so it's kind of laid out like a clock. So I've worked on other projects that have had kind of similar results where we've used this basically as a visualization method to take all this data and kind of boil it down to like, here are the big patterns in our data set. This is probably the most elegant one that I've identified, but there have been some others that have been really useful this way. We actually took this and interpreted it in a number of different ways where we actually were able to extract information out of it. And I'll show you an example of the other end of that with network inference, which is a study we did of mouse response to SARS-CoV-1 infection. This is again from 30 or 40 different transcriptomic data sets where we did a very similar kind of thing to infer networks where each of these is a gene and the links between them. This wasn't correlation, this was using mutual information. So they're basically relationships that were inferred from the data. This is one of those hairballs, right? It's a big mess. So what can we get from this? Well, one, we did clustering. We identified a number of different interesting pathways that were enriched in these clusters. But two, we used topology. And briefly, the topological measures that we used were hubs, and hubs are really simple. It's basically like, I'm in a network, I'm a gene, how many different neighbors do I have? If I have a lot of neighbors, I'm called a hub. And bottlenecks are basically like bridges in a busy city. So in the network, they are places that constrict the flow of information from one point to another. And the really interesting thing we did with this is we said, we're looking for what are the mechanistic, what might be good drug targets? What are the things that are actually causing the pathogenesis of this virus? And so we selected a bunch of these bottlenecks and we tested them in the lab, we, meaning my collaborators, and found that we could identify these really interesting bottlenecks that were actually had significant impacts on the ability of mice to respond to SARS-CoV, just using this kind of fairly simple kind of approach. So that's kind of another story. Okay, almost done here. Several different resources. The string database is a place that collects lots and lots of different kinds of associations, everything from protein-protein interactions to regulon interactions, to other kinds of interactions. It has a really nice interface. You can input your set of genes and it will show you the enriched subnetworks, and kind of you can delve into the relationships between those. It's an open source and free version of something that I put on here because some might have seen it, QIAGEN Ingenuity Pathway Analysis. It's an expensive tool. I haven't used it very much because you have to have a license, and it is primarily targeted at human disease. But I've gathered that it's a very effective and useful tool. KEGG has a nice interface, web interface, where you can, again, this is kind of for the pathway overlay stuff rather than networks per se. Cytoscape is a great tool for just exploring networks, biological networks. It takes a little bit more work to get your networks, but there's lots of plugins to Cytoscape that allow you to download networks and to input your own sets of genes, like that. If you're really interested in inferring networks, there's a couple of tools that we've used quite a bit, and I won't go into the details of those, but important point, but fairly simple. What's important for getting your data ready to do pathway enrichment? Identifiers. So identifiers are always the problem, and this is identifiers of your individual gene, right? So it seems like a simple idea, but oftentimes the experiments that you'll get identifiers out in one format and they won't match up to KEGG database, or they won't match up to something else. If they don't match those pathways, then you'll never get any enrichment because you're basically just doing a big lookup, right, of the pathway gene tables. Most of the tools that I mentioned above, especially the ones with web interface, they will have both things to filter out, like, hey, you've put in a list which we can't match to any of our database. And they also have translation tools. It's still not always an easy process to get your identifiers in the right format for particular pathway databases. And I'd say this is even maybe extra true of bacterial data sets. And especially when you go into metagenomics and you're kind of pushing out into territory that's not well annotated, that can be difficult. Data matrix format, that's rows are genes, columns are conditions, and all of the things that we talked about with data normalization and kind of making sure that your quality is good, also good. So I'll end with a caveat, which is, for a lot of this stuff, we're trying to get it at biological function. Correlation is not always causation. There's a great XKCD that I didn't include here. This is just a really fun website. I think it's still active. Where he basically takes a whole bunch of stats and finds things with very high correlations that are just absolutely ridiculous.